we're excited to welcome you to the Fruits of the North, Innovations in Cold Climate Production. So our experts are going to highlight the groundbreaking research being conducted at the U of M on cold climate fruit production with a focus on apples and grapes. We'll start with an introduction from the Director of Research at the Minnesota Landscape Arboretum, and then have lightning presentations from 4 speakers. Again, we encourage you guys to add your questions to the chat throughout the presentations. We'll moderate a brief Q&A after the speakers finish, and at the end you'll have a chance to interact with our faculty in breakout rooms if you want some more one-on-one time and engagement for your personal questions. So these rooms will be open for 10 to 15 minutes after the program closes, so you can move from room to room if you'd like. We'll have an apple room and a grape room, and Matt will be in a room as well. So it's now my pleasure to welcome Dr. Matt Clark, Associate Professor in the Department of Horticultural Science and the Director of the Minnesota Landscape Arboretum. He's going to provide us with an introduction. Matt leads the fruit breeding program now and directed the grape breeding program from 2015 to 2023. So welcome, Matt.
Well, good afternoon. Thanks, Annie. It's great to be here and to introduce you to some of the work that we're doing. And I guess we could go to the next slide. So as mentioned, today we're going to talk about fruit research and variety development, and also you'll hear from graduate students and staff on 2 different projects who are doing work in this space. So although I represent fruit research, that's actually separate from our grape breeding and enology research program that's led by Dr. Soon-Lee Tay, who came into the program in late 2023 as we had some transitions. And most of you are probably familiar with our apple and other fruit breeding program that was led by Dr. Jim Luby, who retired in August of 2024. And I am the kind of next iteration championing the work that we do on the fruit side. So all of the other fruit but grapes, but today you'll get to hear a little bit about both of those. Next slide, please. So I give a lot of talks all around the country and around the world, and people often say, why apples and why grapes in Minnesota? Why are you trying to do that there? And as we all know, Minnesota winters are very cold. With climate change, we're seeing less deep winter cold, but still have some of the other major problems that we have in our climate with spring frost and major temperature swings. And our growing season is still short because of those frosts. And so it's a challenge to grow fruits and vegetables and flowers and many other crops here. And the key reason that we focus on these crops is that pioneers and homesteaders were moving into Minnesota, and they actually needed to be able to produce food and have sustainable farming operations to be able to claim that land. And the landscape in Minnesota was pretty barren for the crops that they were used to. Of course, we have native grapes and raspberries and strawberries, blackberries on our landscape in Minnesota, but apples and grapes and many other fruit crops that people were used to growing in Europe or in the eastern part of the United States just didn't survive here. And so those crops needed to be developed. I think we take it for granted today that we can see all of these crops in our landscape, and it really has to do because of the plant breeding program here at the university and our long legacy. And I love this quote from Horace Greeley from the New York Tribune. He was a newspaper man who said, "I would not choose to live in Minnesota because one cannot grow apples there." And in 1860, that was true. And in 2025, we have a completely different story thanks to all of the research efforts over the last 125 plus years. Next slide, please. So our hort research started by just regular folks, not necessarily university scientists. The university was developing a horticulture program, but fortunately we had some inventive pioneers who were living on our landscape who wanted to be plant breeders and solve some of these issues. And the first discovery was the wealthy apple. This is Minnesota's first cold hardy apple, which was developed by Peter Gideon. And because of his success, he was hired by the university to become the first superintendent of our fruit breeding farm. At that time was actually on the shores of Lake Minnetonka. And the goal there was to really focus on cold hardy plants and fruit. That initiative didn't last very long. The station was closed in 1889 and was sold for $16,000. And about 20 years later, with support from the Minnesota State Horticultural Society, The state legislature actually purchased the ground where we have our research farm today and appropriated funds for the management of that farm in 1907. Now that we call it the Horticultural Research Center and is part of the Arboretum, and the first plantings went in 1908. Next slide. I love this quote. It's from Charles Harrelson. If you are familiar with apples in Minnesota, you likely have eaten a Harrelson apple. It was released over 100 years ago, named after the second superintendent of the research station. And he said, to get the best results in fruit breeding, a large number of varieties of all fruits adapted to this region should be planted as soon as possible at our new fruit farm. So in 1908, they started in earnest with apples, grapes, raspberries, and numerous other crops. And Dr. Harrelson also said, I didn't put this quote on here, something to the effect of, without the patient and hardworking plant breeder, we won't be able to have the fruit that we need. Because the process takes so long, we have to start today. And so that was his message in 1908. Next slide. So what is plant breeding? At its core, we describe plant breeding as both an art and a science of developing new plants using our knowledge of genetics, genomics, and other fields. In particular, we aim to know as much as we can about the parents who we want to use in our breeding program. That includes their pedigrees, it includes genetic markers, understanding their traits and how they're inherited. And then we make crosses between the parents that we want to use in our program, and then we screen the offspring. And our crews will really affirm that our job is to taste fruit, spit it out, and repeat. And we would taste hundreds of individual seedlings, sometimes every day, as we look for the new varieties, new potential varieties. And so you might ask, how long does it take to create a new variety? And the answer to that is 15 to 25 years for us to go through that process. And to have a huge success, it might take another 15 to 20 years before that new variety really has a big splash in the marketplace. So we're talking about whole careers built around developing these varieties. And pictured here on the left is David Bedford, who's been with the program for, I believe, 44 years. Jim Luby was with the project for 42 years. And the rest of the crew on the Apple project shown here are Kate Skapansky, Sarah Kostick, and Jack Tillman, and myself, all looking forward to the next 25 or 35 years developing new varieties. Next slide, please. So what about impact? Just very briefly, this is just a short list of some of our accomplishments, and you're likely familiar with many of these. We've released 12 grape varieties, Clarion being the most recent, and you're going to hear about a couple of table grapes that are in the pipeline. We have 29 apple varieties. First Kiss and Kudos are some of the more recent ones. We have 5 pears, Juicy Jewel, one that you might be interested in growing at your house, 16 strawberries, 22 plums, and then unlisted here are the other crops like raspberries and currants. So the litany of those plants is long. You might not know that Minnesota has nearly 80 wineries today, representing about $100 million of economic impact to our state and supporting thousands of jobs. The whole cold climate wine industry has really developed out of what's happened at Minnesota and nationally has an economic impact of easily $500 million. And then, of course, just kind of end up with the one that we all know about, Honeycrisp, everyone's favorite apple, is a variety that has a global distribution. It's grown on several continents. And just this past year, I looked up the data this morning, Honeycrisp was ranked 4th in volume for US production. And I think that's it for me. I'm happy to turn it over to our next experts who are going to be introduced. Thanks.
Thank you so much for that introduction to the fruit research that's happening at the U and a reminder of all those varieties that have been released over time. We're just so lucky to have this famous research happening right here in Minnesota. We're excited to hear more from your colleagues. So next, we're going to welcome Dr. Sarah Kostick, who received her MS in her master's in applied plant science from the U of M and her PhD in horticulture from Washington State University. Sarah joined the U of M fruit breeding program in 2021. Thank you, Sarah, for joining us.
Thank you, Annie. Next slide. So I get the pleasure and kind of the huge task of giving a crash course in apple breeding in just 10 minutes. So we'll see what we can do. But before we get into the nitty-gritty of the breeding program, I first want to make sure we're all on the same page in regards to some of the biology of apples. So next slide, please. So if you were to go to the grocery store and you're like, I want to be really cheap about this and I want a Honeycrisp tree, so I'm going to get a Honeycrisp apple, I'm going to extract those seeds, I'm going to plant those seeds, and I'm going to wait a really long time and get that tree. Well, not only are you going to wait a really long time, you're not going to actually get a Honeycrisp apple tree. You're going to actually get an offspring or a child of Honeycrisp. Because why? Seeds are actually the result of sexual reproduction. If you think about how this happens in the orchard, bees are flying around bringing pollen from random varieties to your Honeycrisp tree, and that results in sexual reproduction. Next slide, please. This is easier to see when we actually see what this variation can look like. So what I have is Honeycrisp, and then I have 3 different varieties that resulted from 3 different seeds of Honeycrisp. And these are all half siblings because they share Honeycrisp as a mother and have a different father. But what I want you to take away from this is that each variety results in a genetically unique tree. So one seed results in a genetically unique tree. So you get lots of variation visually, but also when you bite into that fruit. So that if you're going to only take one thing away from this presentation, it's that. Next slide. So how do we consistently get the same variety over and over and over again once we develop a variety? Well, it's through clonal propagation. We literally clone these over and over again. Apples are produced as compound or composite plants where the scion, what we think of as the apple variety, is actually grafted onto another variety of apple, the rootstock, that underground portion. And that's how we consistently produce the same varieties over and over and over again. Next slide. So this may seem pretty straightforward, but we want to think about, when we're thinking about a breeding program, we want to think about the goal. So the goal is to develop new improved apple varieties, stuff that look more like the top and less like the bottom. And you can think about it this way: finding those right genetics and removing the wrong genetics, or looking for fundamentally a needle in a haystack. Next slide. So the first steps in breeding, and Matt kind of alluded to this, is first of all, we want to know what are our breeding targets. What do we want to improve upon? Because if we don't have a target, we're going to not make any improvements. So are we focusing on crispness, juiciness, sweetness, etc.? What are those targets? And then once we have those targets, we can start to pick our parents based on what we've observed, our pedigree information, as well as DNA information. What individuals do we want to combine together? And then once we have those lists of parents, we can start to actually design our crosses. So what combinations that will complement each other. So if we have a parent that's really a delightful eating experience, but it's really ugly, maybe we want to combine that with something that's pretty attractive and maybe doesn't have quite the eating experience, so that potentially we can get an offspring that has the combination of both parents' best characteristics. Next slide. So what are our breeding targets? Well, fundamentally in this program, we are focused on eating quality, eating quality, eating quality. Eating quality is anything you experience when you bite into a piece of fruit. Why are we so focused on this? Well, if you, the consumer, don't have a product that you want to consume, the rest of it's kind of pointless. So eating quality is really important. Appearance, we as consumers, whether we want to admit it or not, buy with our eyes. And so we want something that's attractive and that stands out on that grocery shelf. Storability is really important as well, because apple is stored often for an extended period of time. So we want to make sure that that's maintaining its quality throughout that storage period. And then finally, disease resistance and production traits. These are more at the grower side of things, but we want to make sure that we have disease resistant material as well as good producers so that we can make sure we have sustainable programs. Next slide, please. All right, so now we have our list of crosses that we want to make. We actually have to do this, and we do this through what we call controlled hybridization. And that's really just a fancy way of saying we control what pollen goes on what flowers. Once spring starts to happen and those flowers start to come out, we bag them before they open so that we can prevent any pollinators from getting in there and taking random pollen from different places. We collect pollen from what are going to be our father plants. And then once those flowers are open in those bags, we literally take pollen, put it on our fingers, wipe it on that flowers, bag them back up, and let nature take its full course, resulting in fruit and seed development. And remember, each of those seeds, those are the basis of our breeding program. They're the genetic variation, and each of those are genetically distinct. Next slide. And we actually have to grow these seedlings out, right, in order to be able to evaluate them. So each year we germinate about 7,000 to 12,000 seeds per year. This picture was taken actually a week ago, so this is what they look like right now. And at this stage, I have no idea what this is going to be. Is this going to be the next Honeycrisp, or is this going to be a mediocre or trash apple? Most likely it's going to be mediocre, but we have to have those numbers in order to actually identify that needle in a haystack. But I want to be really efficient with my resources. And so I want to eliminate any individuals, any seedlings that have the highest potential to perform poorly, to be garbage. When we bite into it once, we're not going to want to bite into it again. Next slide. So we do have some DNA tests for specific characteristics. For example, what I'm showing you here is a storage disorder. So we can know, you know what, this individual, based on its DNA information, is more likely to develop this storage disorder than others. And so I can eliminate it before I even have to grow a tree. And so we cull about 3,000 to 6,000 seedlings at this stage before they even grow up with DNA information. What do we do with the remaining seedlings? Next slide, please. Well, we actually have to grow them. We actually have to propagate them and grow them in the orchard. So for each seedling, we propagate one tree. We graft a bud from that onto a rootstock in the orchard, and then we grow those trees up until they start fruiting. Next slide. And this is when the fun begins, when we actually have to go through and taste all of this fruit. So at any given point, we have about 20,000 genetically unique apple trees that we have to evaluate. So come the end of July till about the beginning of November, 4 of us walk each of these rows and look for individuals that are ready to evaluate. And when we determine, you know what, this fruit is ready to evaluate, we do a very quick and dirty evaluation. We're asking a very binary question. Is this garbage? Is this horrible? Or is this interesting and potentially worth evaluating further? And so it takes me like 1 to 2 minutes to evaluate it. Yeah, I'm thinking about firmness, crispness, juiciness, sweetness, tartness, flavor, and appearance, but I'm really just asking that question, is this repulsive or is this worth evaluating further? Next slide. And the answer is usually pretty easy. It's usually, this is either repulsive at worst or mediocre at best. And if that's the case, that's the end of the line for that seedling. I'm going to take out my spray can and spray the trunk orange, and that tree will be removed at the end of the year. And that was that seedling's one shot. It's done. Next slide. But every once in a while, a tree makes me stop. And when I bite into it, I'm like, I actually want to take another bite. This is interesting from a flavor perspective or a texture perspective, whatever that may be. And if that's the case, I'm going to put a flag on that tree. And we're going to come back later in the week as a team and reevaluate it. Because sometimes, yes, it was interesting. I wanted to take another bite of it, but it was just because it was less mediocre than the material I had tasted throughout the rest of that week. But sometimes it's actually interesting. And if it's actually interesting, well, we're going to move that into our next phase of evaluation, where we're going to do more data collection and more evaluation. Next slide. So if something was interesting and we decided, you know what, we're going to select that, we're going to move that from our first phase of evaluation to our next phase. This is really our data collection phase. So we want to propagate, clonally propagate more of these trees. Why? More trees result in more fruit that we can actually evaluate. And we're trying to collect as much information about these individuals as possible from a sensory perspective. So it involves, as Matt said, a lot of biting and a lot of spitting, as well as instrumental data over multiple years, because we want to see the good, the bad, and the ugly about this individual. And until you evaluate over multiple years, you're not really going to see that. And we also look at a lot of storage life. And most of the time, the material in this next phase of evaluation dies what David Bedford likes to call a slow death of mediocrity. And what I mean by this is that it was good enough to get from that first phase of evaluation to this next phase, but it's just average. It's just slightly above average. It's not really any better than anything that's already on the market. And as you know, it's a very competitive market. So if that's the case, if it dies that slow death of mediocrity, it's going to be eliminated from the program. But every once in a while, something performs really well. Next slide. And that becomes our needle in a haystack, our next variety. And as Matt mentioned, it takes about 15, if we're lucky, to 25 years to develop a new variety. So maybe towards the end of my career, we'll have something that I actually designed the cross for. But these are our most recent releases from the program, MN33 and MN80, and they're excellent pieces of fruit. And you should look forward to seeing those come into fruiting in the next few years. Thank you, and I look forward to any questions that you may have at the end. Thanks.
Thank you, Sarah, for that crash course in apple breeding. You really ran through that fast. We're even a couple minutes ahead of schedule, which is great. So we know the apple breeding program is flagship for the U of M. It's really nice to hear more about like how you target things and the process and the outcome. And I'm sure we're all going to look for kudos and triumph at the store. So next we're going to hear from Sam Clayton. a master's student advised by Dr. Matt Clark in the apple breeding program at the U, and they received their Bachelor of Science from UW-Madison in horticulture in 2023. Sam's master's thesis is focused on evaluating chemical tools for key flavor compounds in apples for hard cider. Thank you, Sam, for joining us today.
And thank you for that introduction. So yeah, I will be focusing on a bit of a different flavor and quality trait than for our dessert apple breeding, specifically talking about hard cider apples. Next slide, please. So just a basic question, what is hard cider? Next slide. Hard cider is a fermented beverage primarily made out of apples. Yeasts are used to ferment sugars into alcohol. You can see here one of the airlocks on a cider I made at the HRC, just bubbling away as it ferments. Alcohol contents typically range from around 4 to 8% ABV, and there are a number of styles that exist all across the world. Next slide, please. So we have, cider is also a growing industry here in the state of Minnesota. There are cider producers making ciders out of Minnesota apples, like this Loon Juice Honeycrisp. And there's also a number of local cideries that are making really interesting, great craft ciders. I've listed a few, but there are some really fantastic cideries in the state that you should check out. Next slide. So we have certain apple cultivars that we prefer to use for hard cider. So what are cider apples? Next slide. Put simply, cider apples are just specialized cultivars of apple, just like domesticated dessert apples. I've listed a few English cider apple varieties, as well as MN 1734, which is actually a cider apple that was selected at the University of Minnesota in the 1930s. It never got a name, but it's really popular among cider makers here in the state of Minnesota. Next slide, please. And the University of Minnesota, although we've done a little bit of cider breeding, is interested in doing a lot more of it, specifically looking at cider apples. And unfortunately, cider apples are often really unpleasant texturally and flavor-wise when eaten fresh. And as Sarah mentioned, the taste testing system we do for apples is, do they taste good or not? And none of these taste good fresh. So that system doesn't really work particularly well. We could also go about the fermentation route. It's a fermented product, so going through that would be helpful for figuring out final flavor. But unfortunately, it's a really time-consuming process. We just don't have the time or resources to do hundreds of fermentations. So my research is looking at chemical testing early in the evaluation process, kind of in that first test phase, that would allow us to more efficiently make selections. Next slide, please. So when it comes to cider flavor and the flavor of cider apples, bitter is often better. Next slide, please. And I promise this is the only organic chemistry I will have in my slides, but I promise it's really important. So phenolic compounds are the chemical compounds in apple that provide both bitterness, which is that flavor taste, or that flavor or taste, and astringency, which is a sort of mouthfeel. If you've ever had a glass of red wine, that mouth drying sensation is astringency. Predominantly, this is from flavanols, but there's also compounds called hydroxycinnamic acids that are important in other ways. And you can see they all have that ring structure there with the OH groups, and that's what makes them phenolic compounds. Now, if you take phenolic compounds and squish them together, next slide, please, you get tannins. Tannins are just condensed flavanols, and the longer those chains you smush together, the more astringent and less bitter you get. So you get more of that pure sort of mouthfeel as opposed to flavor. Shorter chain tannins and those low molecular weight compounds are more just bitter, which is still important, but just different. Next slide, please. So tests from the wine industry have provided me some options for measuring bitterness. I won't go in depth, but basically we have a test that measures total phenolics, so those long and short chains, and also just tannins. Next slide, please. So we surveyed phenolic content in a range of more or less relevant apple varieties as potential parents for the breeding program, specifically looking at a lot of English and French cider apples that are cold hardy, as well as in bold apples that were developed at the U, a few ornamentals, and some wild apples. Next slide, please. Next, oh, there we go. So looking at the results, how do total phenolic and tannin concentrations vary in our in our varieties that we studied? Next slide, please. Looking at total phenolic content, we can see a really wide range between varieties, ranging from those dessert apples in green, I would say are just straight up not bitter. There's really no detection, to in the blue, those wild or ornamental varieties, a flavor profile I can only describe as poisonous. Just deeply, deeply unpleasant and not something you'd really even want to make cider out of in large amounts. But you can see in red, most of our cider apple varieties are within a similar range of one another, higher than dessert apples. Next slide. Looking at tannin more specifically, it also varied really widely in our population, ranging from, in the case of Zestar, literally no tannin, to a decent amount of tannin. And you can once again see that those those cider apples are really where you find those tannin molecules. Next slide, please. So thinking about some key takeaways. Next slide, please. Using chemical tests, we can more efficiently make selections for apples that taste really unpleasant while eaten fresh and save Sarah and other researchers some really painful and unpleasant tasting experiences. Next slide. But additionally, to produce high phenolic ciders, we just need to be using higher phenolic apples. Dessert apples, while they can be really tasty, just aren't going to produce the flavor profiles you need like a bittersweet or a bitter sharp cider apple will. Next slide, please. And finally, my research has found that the University of Minnesota apple breeding program has some really great options for potential parents of crosses. We have a wide range of phenolic content in some of the apples at the HRC, and we have a lot of options for making some really cool crosses. And that's all I have.
Cool. Thank you so much, Sam, for sharing all that work studying the cultivars and selection methods for the hard cider industry. I'm sure that many people on the call, including myself, look forward to the advancements in hard cider, especially as we approach patio season. I'm personally a milk and honey heirloom cider person. If you haven't had it, give it a try. You can also visit the location out there in St. Joe, which is a fun little field trip. So next we're going to switch gears. We're going to discuss grapes. I'm going to welcome Dr. Erin Treiber. She's currently a table grape researcher in the grape breeding and enology lab. She's worked with the team for 7 years. She completed her PhD in plant biological sciences at the University of Minnesota. Thank you, Erin, for joining us today.
Thank you, Annie. I'm really excited to talk about some new Minnesota table grapes today. Next slide. So as Matt mentioned briefly, our program has released 12 grape varieties. I've pictured some of the wine varieties we've released, which are lovely. We have only released 2 table grapes, and the last one was in 1977. So as someone who focuses on table grapes, I think we're ready for some new ones. Next slide. So I'm going to focus my talk on 3 new advanced selections that we're really excited about that are in the pipeline for release from our breeding program. The first, and this has gone through a long process, like Sarah talked about with apples. So this is the end of a lot of work. So our first grape, I'm going to refer to them as their color, is a green grape. It gets actually a golden color when it's ripe. It has a beautiful pineapple tropical flavor. It also has a similar aroma. So when you walk through the vineyard, you can smell this grape and you know it's ripe. This grape, along with the other 2 I'm going to introduce, are virtually seedless, which means that they're mostly seedless. They sometimes have a trace or a little bit of a crunch when you eat them, but they're nothing like a seeded grape. They're more on the seedless side. The next grape is our red grape. This grape is fruity, it's strawberry, it has a mild flavor, it's a really lovely grape. And then finally we have the black grape, which this one is crunchy, it also has some fruity notes, and it has a unique flavor. I joke that this grape, you either really like it or you don't really like it at all. There's no really in between. We also jokingly call this the blueberry grape. It has a similar texture, color, and size to blueberries, and we've actually baked with them similar to how you would use blueberries in the program. Next. So some current and ongoing projects, these are under provisional patents, and they're being grown by several test growers in Minnesota and Wisconsin. And we did evaluate some consumer liking at a farmer's market. And I'm going to focus in on these last 2 in the next few slides. Next. So first of all, I would like to introduce, we had some funding from the Minnesota Department of Agriculture, and I was able to work closely with 3 growers in Minnesota to start growing table grapes. The first of them is the Freund Farm up in Isanti, Minnesota. Tom and Kay Freund are a small vegetable farm. They grow mostly asparagus and rhubarb, and they sell right out of their farm. So they had never grown grapes before this project. On the other end, we have 3B Vineyard out in Dawson, Minnesota, mainly working with Holly Thompson. Their family actually they're traditional row crop farmers, but they also grow wine and table grapes. So Holly was kind of our grape grower already. She does farm to school with her table grapes and also makes juices and jams. Finally, we have Erin Wills down at Little Hill Berry Farm in Northfield, Minnesota. Erin runs an organic pick your own blueberry farm. He also has strawberries and some raspberries and a few other things. And it was his first time growing grapes as well. Next slide. So through this grant and a partnership with the Minnesota Farmers Market Association, we were put in touch with the Riverwalk Market Fair down in Northfield, who offered us a booth for a morning in September. Erin's farm is near Northfield, so he was able to come with us, and we had some of these grapes for people to taste. We wanted to talk to people, see what they thought. We also did a written survey. We have Minnesota Nice, so we knew it would be hard for people to maybe say something negative about the grapes to us. So we had a short survey that we had people fill out. It was only 3 questions. And I'm going to share a little bit of the results of that survey with you next. Next slide. So we did get 103 responses. If anyone's ever had to get survey data in person, it can be difficult. So we were really excited to talk and get 103 people to fill this out. And our first question was, how do these compare to grocery store grapes? And the responses could be better than, equal to, or worse than grocery store grapes. So for our green grape, 85% of people thought they were equal to or better than the grocery store grapes. And actually 68% of those said they were better than grocery store grapes. The red grape had 96% of people saying they were equal to or better than grocery store grapes. And 73% actually said better than. And then finally, our black grape, 82% found it equal to or better than grocery store grapes, with 55% of people saying better than. Next slide. So then we asked people, which grape was your favorite? So out of the 3 you tasted, which one did you like best? And then we asked them to describe what they liked about it. So our most popular grape was the red grape, with 46% of people choosing it as their favorite. They said it was sweet, crunchy, it had fun flavor notes. Someone said it had the perfect sweetness. Next was our green grape with 38% of people, and they described it as sweet, delicious, slightly tart, and someone said it tastes like candy. Finally, our black grape, 16% of people still like this. Like I said, it's a bit polarizing, but what they did like about it was they liked the flavor and texture. Some people found it sweet. They said it had a unique flavor, and they found it slightly more tart. And hopefully with these data and sharing with you, you might be asking yourself, where do I get these? I'm actually really excited. Next slide. Well, unfortunately, grapes do take 2 to 3 years to bear fruit, and these are still in the patent process. So the earliest we'll probably see these ready for a harvest in about 4 years. Once they come out, though, you can look for them at pick your own farms, farmers markets, and probably local food hubs. However, until then, next slide. Please join us at our September field day. I always feel bad. I get to talk about these great grapes, but you don't get to taste them. The first week of September every year, we hold a field day where you get to taste over 50 varieties of grapes that our crew collects. It's open to anyone, and it's a great opportunity to really get to taste a lot of these Minnesota varieties. I put our website underneath the picture so you can follow and see, kind of check on the details of that. I also just want to mention Soonli Tay is the leader of leads our program, and he would be the one, if you're really interested in growing these, you might want to reach out at tay@umn.edu. Thank you for your time.
Thanks, Erin. Thank you for sharing your work in the new table grape varieties. So as a former farmhand who did some work back with Matt at the HRC in 2015, I can attest to these varieties and their delightfulness. I like the black grape personally. I'm excited to watch for these varietal releases, and I will put the September field day on my calendar. I hope many of you on the call will as well. So now I'd like to welcome our final speaker, Doug Vines, a master's student in applied plant science at the U of M, advised by Dr. Matt Clark and Dr. Soonli Tay. His master's thesis focuses on different methods for quantifying cold hardiness in hybrid grapevines for genetic and phenotypic assay development. Now, I know that's probably a very, very important part of the cold hardiness portion of the grape breeding program. So thank you for being here, Doug, and rounding us out.
Of course. Thank you so much for the kind introduction. Again, we're going to be talking a little bit about cold climate grape breeding. So next slide. I'm sure as alumni, y'all know that Minnesota winters are cold most years. Last winter was a little bit different, but all in all, the winters in Minnesota are too cold for the domestic European grapevine, Vice vinifera, to grow up here. So that means, next slide, if we want to have a grape industry in Minnesota, we have to grow hybrid grapes, which get their cold hardiness from wild relatives. In our breeding program, we mostly use Vitis riparia and Vitis labrusca as cold hardy parents. I'm going to focus on Vitis riparia because it's native to Minnesota. So this picture I have of Vitis riparia growing up a tree, I took that on Nicollet Island. And the reason we just don't grow Vitis riparia as is because one, it grows like a weed. And two, you can see this picture here, it's sour, it's small, and it's seeded. We don't want to eat that. So as we grow new cultivars and we develop new cultivars, we want to make sure we're keeping that excellent cold hardiness from our wild parents. So next slide. In order to keep that cold hardiness, we need to know how to evaluate cold hardiness. In the past, we've done this with bud chops, which can give us an estimate of winter bud survival. So this image here is a cross section of a grapevine bud. Within a grape bud, it's a compound bud. So there's a primary, secondary, and tertiary bud. And this is important because grapevines, like other woody perennials, all of the new growth lives in this dormant bud that overwinters. So this image, you can see the primary bud is brown, it's dead, which means it won't grow. The secondary bud might take over, but it won't have as much fruit. So this is a problem. We do this for growers. We can give them a percentage of bud survival, but as researchers, we want to know when the bud dies and at what temperature the bud dies. So we need to look at this in a different way. Next slide. And we're interested in when and why and what temperature the bud dies because grapevine hardiness is dynamic. I've modeled this here with our daily high and low temperature up top in Chaska, Minnesota in the 2023-2024 winter. At the bottom, I've got what the grapevine is doing. And all grapevines, they have this sort of U-shaped curve where they acclimate in the fall, they gain cold hardiness. In the deep depth of the winter, they maintain cold hardiness. In the spring, they deacclimate and lose cold hardiness. While all of our grapes have this conserved kind of U-shaped curve, they go about it in different ways. Some grapevines might acclimate faster, slower. Some might maintain cold hardiness at different temperatures, and some might deacclimate faster or slower. So we're interested in seeing those differences. Next slide. The way we go about this is in differential thermal analysis, or DTA. What I do for this is I go out every week, October through March, no rain, but snow or shine, and I collect the dormant wood. I take this wood inside and I cut the dormant buds off and I put them in these trays, which have these wells that have really sensitive thermometers in them. We put the buds in those wells inside of our fancy freezer. It goes from 32 degrees freezing all the way down to -40 over the course of about 12 hours overnight. When the bud freezes, it releases just a little puff of heat. Our temperature sensors and our wells can capture that, and we can track at that day which cultivars died at what temperatures. So I'm sure you're all really interested in seeing these results. So next slide, I've got you some from the 2023-2024 winter. And what we saw first is that variation is present in our grapevine hardiness. I'm showing you all 12 different cultivars. These are mostly wine grape cultivars with our black, green, and red grape. What we see is there's some variation in acclimation in October and November. There's some variation throughout the depth of the winter, but there's a lot of variation in our spring, how things lose hardiness and deacclimate. Now, this looks like a bunch of rainbow spaghetti, so we can pare this down into our main patterns that we see on the next slide. Thank you. So I picked Itasca, Marquette, and our red grape, that's our red table grape, as examples of our 3 hardiness patterns. I'll start with Itasca in blue. It acclimates quickly, it gains hardiness quickly, and then it maintains a low deep winter hardiness. Then in spring, it deacclimates slowly. It loses hardiness slowly. Marquette's our second pattern. Similarly, it acclimates quickly. It maintains a low deep winter hardiness, but then it deacclimates and loses hardiness quickly. Then we've got our red grape. It takes a little bit longer to acclimate. It takes more time, and it maintains just slightly warmer than our Itasca and Marquette groups, and then it deacclimates quickly. So next slide. We've got some final takeaways from my study. What I want you all to learn from this is that while dormant hybrid grapes, like other hardy woody plants, respond to their environment, even though they look like they're not doing much, they're doing quite a bit. And we've found that there's a lot of variation in the patterns of our hardiness on our grapevines, and we've found 3 patterns of hardiness that can help us select for better hardiness in the future. Thank you all so much.
Wonderful. Thanks, Doug, for teaching us more about the work in evaluating cold hardy vines and for vines for wine and table grapes. So thank you to all of our speakers. We've really covered a lot of ground here today. We threw a lot at you guys, all about the fruits of the north. We hope you found it valuable. I'm sure interests are piqued. I know we've had some questions come in through the chat, but if you are interested, we are opening it up for questions from the audience. So we have had some come in through the chat, but if you want to unmute yourself and ask a live question, we'll open that up first. And if we don't get any live questions, we'll lean on our Q&A in the chat. But please feel free to come off mute and ask a question directly to one of our speakers if you'd like. Anybody brave enough?
Yes, I'd like to ask a question. Do you evaluate resistance to disease and the ability to be grown organically when you breed cultivars of all these different types of fruits?
I guess I can answer that one or take a stab at it. So we are certainly thinking about organic production where it makes sense. The climate in Minnesota, with the amount of rain that we receive typically in the summer, does make it quite a bit of challenge to manage some of the diseases, especially on the grapes, that are impacted easily by about 5 different fungi. Apples is a slightly different story, where there's one major fungal pest and one bacterial pest that are problematic. Big problems in Minnesota, not a problem in Washington, where it's nice and dry. They don't get the same types of diseases. But we do have our eye towards sustainability in all of our breeding programs. And so one of the benefits of using some of the wild species in our breeding programs is that we can select for disease and insect resistance. It takes time. Crossing into wild species brings in a lot of bad traits that we have to select against. So it adds to our clock of how long it might take to release something. But the example of the cold hardy grape, not only did the species that Doug mentioned bring in cold hardiness, they also brought in new flavors, pest resistance as well.
So I have a comment to make, which you might find kind of off the wall, but I'm a big tomato grower, and of course, tomatoes are susceptible to fungal diseases. And I was on a farm tour with the co-ops, and we stopped at an organic farmer's homestead. And he had a bunch of tomatoes, and he'd put a hoop house over them, open at both ends. So these tomatoes never actually got any rain. They were irrigated, drip irrigated. They had, this was like August, they had absolutely no fungal diseases. And I'm wondering if you adopted this procedure of just covering the plants and giving them drip irrigation, if they would be much less susceptible to fungal diseases. And just throwing it
out. My gut reaction, and there's probably other scientists on this call, but a lot of the fungal diseases in tomato are rain splash dispersed in the same way that apple scab might be. So drip
that's true. That's true, but also if the leaves never get wet.
Yeah, exactly. Yeah, so being underneath the canopy could prevent something like that. It would be great if you were a marketer of hoop houses to be on this call and say, we can sell you acres and acres of protection. But yeah, it does present some unique challenges and opportunities using structures like that. Yeah, good anecdote. I saw someone raise their hand. There it is. Yeah. William?
Yes. So I've been hobby growing apples for over 50 years, and I learned quite quickly that in order to get decent eating apples, you need a significant insecticide spray program. And pretty early, for plumper crulio, apple maggot, codling moth. My background is in ag biotechnology. And I know that there are BT genes that have been approved for each one of those 3 different insects, for the coleopterans, the dipterans, and the lepidopterans. So why aren't we developing genetically engineered BT-resistant apple and significantly reducing the amount of spraying that we need to do on apples?
I think the answer there really comes down to consumer acceptance, as well as regulatory hurdles. and the costs associated with them. So using gene editing or GMO techniques are available in many crops. But markets drive a lot of the research we do in variety development, and we haven't seen, at least in the United States and certainly not in Europe, a push for adoption of those types of crops. That's one. And then two, it's really, really hard to do those in apple. It's hard to get apple into tissue culture, especially the varieties we're interested in, callus culture and tissue culture, which might be a little deep conversation for some people on the call, but those plants are just really recalcitrant to those techniques. And so we do have researchers here at the university and across the country who are working on how to optimize those procedures, but the efficiency is extraordinarily low compared to some of the other cropping systems.
So one point, University of Minnesota is the elite educational institution in the state. I'm sure that you know that genetically engineered crops are safe. As a matter of fact, there has not been a single documented case of any harm from genetically engineered crops. Don't you think you have a responsibility to educate the people? The second point is I just talked with Dr. Feng Zheng in the College of Biological Sciences, and he is able to transform by directly transforming meristematic tissue and can select branches and so forth that have been transformed. So the technology is there. All it needs is the education on the part of the university to convince people that they're as safe as they really are.
Yeah, thanks for your comments.
Any other questions from the audience?
I see a hand from Kristen. Oh, but we don't hear you. You might have to put your question in the chat. Or maybe it's already there. It's already there.
Yeah, I see Kristen's, so I think it's about, is it possible or advisable to try to clone a table grape onto this rootstock? And alternatively, should we just dig them up and start over? And then finally, what variety do you recommend? And should we wait for new varieties? So I have to be honest, so in Minnesota, we don't have to use rootstocks. So that usually has to do with phylloxera issues, which is a pest that decimated Europe. The great news is the native vines are resistant to that, or they tolerate it. So we don't do much with rootstock. So I can't really speak to that much. I would say if you don't have to use a rootstock, don't. So I would just dig them up and start over. Or if you have room, expand. The hard part with perennial crops is that you usually want to keep them for a long time. So as someone who, of course, wants you to grow our new varieties, I would say wait. However, if you're really excited to get something in the ground, I feel like the most commonly grown grape here is that's not Concord-like or tastes like Concord, is called Somerset Seedless. It was actually a Elmer. Oh my gosh, I just dropped the name. Matt, you might want to save me.
Elmer Swenson.
Swenson. His varieties are called Swenson, I should know. So he developed Somerset Seedless. It's pretty much the same. It has that little trace. It's virtually seedless. Actually, the red grape we've developed, I feel like, is a little bit of a improvement on Somerset Seedless, but it's also a wonderful grape. So if you want something really, really hardy, you can go with things like Bluebell or some of those Concord-like varieties where you don't get as interesting or unique flavors, but you're going to get a lot of cold tolerance.
Thank you. Now I can talk. So
Awesome.
thanks very
Perfect.
much. All right.
You're welcome.
Okay, in that same vein, similar, I guess, somebody who lives in the Minneapolis-St. Paul area is asking about, maybe, do you know the context for this one, Erin? When are the best times to apply? Oh, is that related to the question prior? Chemicals safe for bees to prevent worms and other
that's the
insects
question
and apples?
it's related to the question
Okay,
before.
and best times to apply. So that's actually a Matt question.
Sorry, I think I missed what the question was.
That's
though.
okay. It was a little convoluted. What chemical can I use that is safe for bees to prevent worms or other insects in my apples? And when is the best time to apply for someone who lives in the Twin Cities?
Yeah, that's a good question. Your best bet is actually to avoid when your pollinators are out if you're going to apply insecticides. And so bees are active primarily in the day when it's warmer. And so that's usually the recommendation that Extension gives, is to target your insecticides in the evenings. And also, the bees aren't going to be visiting your apple trees outside of bloom. They have better interests visiting other flowers for their nectar and their pollen. So around bloom time, of course, is a good target for dealing with some of the pests that someone mentioned earlier. But waiting until petal drop is a great strategy to make sure that you're applying those insecticides when the bees aren't there. So I don't have a chemical off the top of my head to suggest, but those are just some good strategies to avoid your pollinators.
And Matt, how much does the U get for royalties from the various fruit varieties that are released? Might be of interest to many on the call, just out of curiosity.
That's a really good question. And I work in this space spending like 10 plus hours a week, and I actually don't know that answer. But I can tell you, most of our fruit crops, the grapes and the apples, we do collect a royalty on the tree or the plant. And that ranges between, depending on the variety and the mechanism, 25 cents to around $1. And that royalty might be charged to you as a homeowner who's buying that, or even commercial growers who are putting in hundreds, if not thousands of plants. But I'm kind of embarrassed that I don't know the answer of what that is.
That's all right. If you find out, we can figure out a way to get the information out to the rest of the group here on the call.
Matt, do you mind if I?
Yeah, go ahead, Sam.
I was just going to say one data. I have an individual data point here, but for a little bit of context, some of my research is funded through, I think, primarily through royalties from the program. So I don't know an exact number either, but at least enough to fund at least one graduate student.
Perfect. That's a great segue. I was just going to say that the funding into the different programs does come back into the program itself through at least one channel, which does allow us to pay some of the staff on the call and graduate students. And it does vary on market conditions. And as you know, patents do expire, so we lose control of those at a certain time.
Sarah, there was a question for you on who gets to name the fruits and how do you choose the names?
Not me. It depends on the fruit, and it usually goes through our commercialization office. So that Matt can probably answer this better than I can, but the breeding program itself, at least the staff do not name the variety. Matt, do you want to add?
Yeah, we have a number of different processes. And so our Office of Technology Commercialization is actually working on a document that describes the many different ways this has occurred. So some varieties, we've opened up a survey to within the college and people provided suggestions. Sometimes we go back to that well or to other lists and say, oh, what was in our top 10? And does that name work with our new variety? But we will joke that it's the hardest part of this whole process because we can come up with our best names and then we send it to the lawyers and they say there's absolutely no way, or there's already something trademarked in our category, or that word does not translate well into another language. It means something way different than what you think. And so our names that we come up with get shot down, and then so far, I guess we've been pretty successful, but it's problematic. And a really challenging step.
Erin, in that same vein, do the 3 new grape varieties that you mentioned have names? Are you working on that?
I think there's some ideas that the Matt and Sulie have kind of been throwing around, but there's nothing that's certain right now.
Okay, and another one for you. Where can a home gardener find an edible Minnesota hardy grape to plant in their yard? And do you have tips on how to plant them or where?
So generally you can find them at any store you can get plants. You just kind of have to look for them. I found that nurseries tend to have a little bit more variety and a few different ones. I would say, and I think all of them are great. We do have a lot that are out there that taste, I keep going back, but a lot of them taste like Concord. It's a common flavor that shows up. Somerset Seedless, like I said, has a little bit different flavor profile. And I would say just plant them early. You want to make sure you're past frost, but plant them early. John Toole, who is our vineyard manager, likes to say grapes don't like their feet wet. So you really don't want to overwater grapes. We give them a little bit of irrigation when they're first planted and they're young vines, but as soon as they become older, they really don't need any assistance getting water. They have very strong root systems. And then also just making sure they have a great place to get sun and there's good airflow. Like Matt mentioned, like there are some fungal diseases that are problematic, and the better airflow you have, the more that can kind of air those out. And if someone really wants to grow them and has additional questions for me on that, I'm happy to answer those via email too, if we don't get to it today.
I also wanted to jump in on our enology website. There's some information about growing grapes. It's enology.umn.edu. And I just put it in the chat for everyone.
Thank you, Doug.
Okay, and I will remind you guys, we will have a chance to have breakout rooms if you guys do want to have a little bit more one-on-one time, and you can hang out a little bit past 1 o'clock here. You can pick the brains of our researchers and our scientists on the call here. So firstly, before we break out into those breakout rooms, I want to turn the program back over to Mary Bichetti, who is the Director of Alumni and Constituent Relations, and she has a couple of brief comments about some things we have coming up.
Hello, everyone. Thank you to all of you who took the time to be a part of this Classes Without Quizzes program. As Annie mentioned, this started in 2002, and it's just a really great way to highlight our faculty, staff, grad students, researchers that are doing work here at CFANS. I just wanted to remind you all that there are a couple of activities coming up that I hope that you can join us in if you're here locally. Borealis Night of Excellence will be on the 17th of April on the St. Paul campus, and there'll be an announcement going out today who the 2025 Borealis Award winners will be. Also, for any golfers, I hear some golfers say that Topgolf isn't really golf, but I also hear a lot of folks say that they really love Topgolf. And so if you'd like to help, Amy Alt is on the chatted here that she's a development officer. If you'd like to help Amy and the Alumni Society raise funds for student scholarships, the Harvey Jones Topgolf for scholarships will be on May 20th. We love matching our alums and friends of the college with undergrads, grad students, and postdocs. So if you haven't done it yet, or you know somebody who'd like to be a mentor, let us know. We'd be glad to get you connected. And finally, be sure when you go to the Great Minnesota Get Together that you stop by the CFANS booth in the Ag Hort building and visit CFANS, check out our display this year. So thank you all on behalf of CFANS for being here. Taking it back to you, Annie.
Okay, thanks, Mary.